DNA sequencing is the process of determining the order of nucleotide bases in a strand of DNA. In this video, we're going to cover sequencing DNA using Sanger sequencing. So let's say I have my template DNA that I'm curious to know the sequence of. To do that using Sanger sequencing, I first need to make lots and lots of copies of it in almost the same way DNA replication works. To start, we add heat to denature the double strands into single strands. Then we add primer, a short strand of DNA that's complementary to the 3' end of the template strand and acts as a sort of starting position for polymerase to start growing the strand. In this case, our primer is radiolabeled so we can detect the DNA on the gel later. But side note, sometimes people use radiolabeled nucleotides instead of a labeled primer. We need DNA polymerase, obviously, and then we have all 4 deoxynucleotides, adenine, cytosine, thymine, and guanine. Once the polymerase gets going, it elongates the chain by incorporating deoxynucleotides that are complementary to the template strand. And finally, the secret ingredient of Sanger sequencing, dideoxynucleotides. These are slightly modified deoxynucleotides, and to understand them more, let's have a closer look at how deoxynucleotides are incorporated into a growing strand. Deoxynucleotides have a structure with a ribose sugar, a nitrogenous base, either guanine, adenine, cytosine, or thymine, and 3 phosphates. The phosphates are connected at the 5' carbon of the sugar and a hydroxyl group at the 3' carbon. When undergoing elongation, the 3' hydroxyl group is like the glue. The 5' phosphate of an incoming nucleotide binds to the strand by forming a phosphodiester bond with the 3' hydroxyl group of the nucleotide in the growing strand of DNA. Sanger sequencing takes advantage of this by using a specific type of nucleotide called a dideoxynucleotide. You can see that the dideoxynucleotides are similar to normal nucleotides with one key difference. They lack a hydroxyl group on the 3' carbon of the ribose. A tip to help you remember, deoxy means lacking oxygen, so dideoxy means lacking 2 oxygens. Now, lacking this hydroxyl group is a big deal for DNA replication and elongation, because without it, any incoming nucleotides won't be able to form a phosphodiester bond, which means no nucleotides can join the strand, which means that's the end of elongation. The strand won't be able to grow anymore. But why, you might be asking? Hang on a sec and I'll tell you exactly why. To recap, in each reaction mixture, we have lots of template DNA, primer, polymerase, and all 4 DNTPs. Finally, in each reaction mixture, we add one type of dideoxynucleotide in small amounts. So 1 mixture of adenine, 1 of guanine, 1 of cytosine, and 1 of thymine. We'll leave the mixtures to react. The DNA polymerase does its job in elongating the complementary strand using the 4 deoxynucleotides. Then suddenly, a dideoxynucleotide gets picked up by the polymerase and incorporated into the strand instead. Because there's a hydroxyl group missing on that last dideoxynucleotide, the phosphate group from any incoming deoxynucleotide can't form a bond, so elongation for this strand is thus terminated. This is why Sanger sequencing is also called the chain termination method. So polymerases don't really care if they're using a dideoxynucleotide instead of the normal nucleotide. If the base is the right complementary one, it'll add it. That's why we end up forming fragments of different sizes. For example, in this mixture with thymine dideoxynucleotide, wherever a thymine base belongs, polymerase randomly adds either a normal thymine nucleotide or a thymine dideoxynucleotide. The dideoxynucleotide, of course, ends elongation, so we end up with 2 lengths of strands, 6 bases and 7 bases. Using this dideoxynucleotide method, we're going to know exactly what the base at the end of each strand is. This will allow us to piece together the sequence one by one like a puzzle. So we'll run each of the 4 reactions on different lanes on an agarose gel. And when the run is complete, expose the gel onto film. The radiolabeled primer we added in each mixture earlier allows us to visualize the strands. We can now see the strands on the gel and determine the length of the strand and the last base added. The smallest strands move through the gel the fastest, so let's start figuring it out from the bottom up. The shortest strand is 1 base, and we know that is adenine because it's in the adenine lane. The 2 base strand is a guanine, then cytosine, and so on. Then working backwards, knowing adenine pairs with thymine and guanine pairs with cytosine, we can finally decode the template strand. So this method is pretty old school, but it's important to understand the concept because we still use dideoxynucleotides today. For this modern Sanger sequencing technique, instead of fragments being radial labeled, we use fluorescently labeled dideoxynucleotides to detect them. Each of the 4 bases is a different color. Then, instead of using an agarose gel, we use capillary gel electrophoresis. The fragments run through the capillary and are separated based on size, the shortest running through the gel first and the longest last. The fluorescent dideoxynucleotides are excited using a laser. The light sensor detects this emitted color, and a software plots the intensity on a chromatograph. Now we know the length of the strand and the base which was incorporated last, so we can determine our DNA sequence. This new method of using fluorescent dideoxynucleotides made everything a lot faster and cheaper, also considering that you didn't have to make separate reaction mixtures and could just throw everything into the same mixture. And that's Sanger sequencing. For more videos on DNA sequencing, check out my channel and please like and subscribe if this helped you out. Thanks for watching.